This pictogram shows the collapsed beginnings and ends of every intron in the human genome. The height of each letter reflects how commonly this nucleotide appears at this position of the exon-intron junction among all human splice sites. The GT at the donor and the AG at the acceptor are almost invariant. These are called the essential splice site. Based on sequence conservation information, Genie defines the donor extended splice site region as spanning from the fourth to last nucleotide of the exon to the eighth nucleotide of the intron, encompassing 4 windows of 9 nucleotides. The extended acceptor region is comprised of the branch site, the polypyrimidine tract, and the acceptor zones. Within this, Genie defines the acceptor zone as spanning from the ninth to last nucleotide of the intron to the second nucleotide of the exon, encompassing 3 windows of 9 nucleotides. For the branch site zone, Genie uses 4 sliding windows of 6 nucleotides. For the polypyrimidine tract zone, Genie assesses 6 sliding windows of 8 nucleotides. As an example, let's look at the intron 13 donor of the PKD1 gene. Here is the donor essential splice site, here's the donor extended splice site, and here are the 4 windows of 9 nucleotides. Splice site strength can be sensitively measured based on how commonly a 9-nucleotide sequence occurs at this position of the exon-intron junction among all human donors. We call this measure native frequency, or NIF. NIF represents the variety of human splice site sequence combinations that are known to allow splicing to occur. For the PKD1 intron 13 donor, the sequence in the E-1 window occurs at this exact same position in 317 other introns in the human genome. The E-2 window is in 601, the E-3 window is in 592, and the E-4 window in 172 other introns. To convert these NIF raw frequency counts into a score on a 0 to 1 scale, we use a cumulative frequency distribution percentile scale. Let me explain what this is. There are around 250,000 human introns, each with donors and acceptors. The NIF percentile scale ranks donor and acceptor 9-nucleotide sequences from the least common to the most common. Each decile of this graph contains around 25,000 donors, and each percentile around 2,500 donors. The right-hand y-axis shows the raw NIF frequency or count. Of the 262,000 possible 9-nucleotide combinations, only 6,702 sequences for the E-3 to D+6 window actually exist. The least common donor sequence has a NIF of 1, meaning it occurs at 1 intron in the human genome. There are 3,762 unique E-3 to D+6 sequences, and all of these comprise the first and second percentiles of the NIF percentile scale. Whereas the most common donor sequence has a NIF of 2,964, present at the same position of 2,964 different introns, and alone comprising the 99th to 100th percentiles. So, if we look at the least common 10% of all donors, the raw NIF frequency ranges from 1 to 29, containing around 5,700 different 9-nucleotide sequences, each unique or very rare. These rare splice site sequences have evolved to need help from splicing enhancers to work efficiently so that gene expression can be regulated. And if we look at the most common 10% of donors, the raw NIF frequency ranges from 1,741 to 2,964, with only 12 different 9-nucleotide sequences in this decile because each of them are so common. These common splice sites have been evolutionarily honed to be easily recognized and bound by the spliceosome. Therefore, they work efficiently. The remaining 80% fall within this plot. So, going back to our PKD1 intron 13 donor, for the E-4 to D+5 window where U5 binds, a raw NIF frequency of 172 lies at the 34th percentile of a NIF percentile scale. The NIF frequencies of the two central windows, where U1 binds, are averaged, with an average NIF of 597 corresponding to the 62nd percentile. And for the most intronic window, where U6 binds, a NIF frequency of 317 lies within the 54th percentile. Similarly, let's look at the ABCA4 intron 45 acceptor as an example. Here is the acceptor essential splice site. The acceptor zone within the wider extended acceptor splice site region spans from the 9th to last nucleotide of the intron to the 2nd nucleotide of the exon, divided into 3 windows of 9 nucleotides, with the raw NIF counts being 113 for the A-7 to E+2 window, for the A-8 to E+1 window is 396, and for the A-9 to A-1 window, 528. These correspond to percentile scores of 0.46, 0.80, and 0.93. So in terms of how common a donor can be, the PKD1 intron 13 donor is fairly average. And in terms of how common an acceptor can be, the ABCA4 intron 45 acceptor is above average. As typical or above average splice sites, they can be easily recognized and easily used by the spliceosome.